Princess Julapon Science High School Buriram. Next, Oyita Prefectural Mysuru High School. Next, Sapporo Kaisei Secondary School. Next, Chawat Vidyakan School. Next, Princess Julapon Science High School Mukdahan. Next, Seishin Kakuen High School and Junior High School. Next, Princess Julapon Science High School Shunburi. Next, Tennoji High School attached to Osaka Kyoiku University. To begin the section, we would first like to explain the guideline for each presentation. The time for each presentation is 15 minutes. 1 minute for the preparation, 7 minutes for presentation, 5 minutes for the comment and discussion, 2 minutes for certificate and take a photo.
Please check the time show on the monitor over there. If you show this sign, it means that there 1 minute left. And if you see this sign, it means that you should stop because the time is up. At the end of the oral presentations, every presenter will receive a certificate of presentation, and we encourage everyone to come together in your respective for your group photo sessions. During the presentations, please switch off your mobile phone if you want to take photos. Then please stay in your seat and refrain from walking around. Be mindful of the audience.
Drinking and water snacks are served on the table beside of the classroom number 2301. And certificate you can download at TJSFF 2023 website.
The presentation begins at 9:30.
test. Okay. Ohayo gozaimasu and good morning every participant and commentators. My name is Eklin Panyo.
My name is Poipat Minisorn Chumsin.
We are grade 12 of Princess Churaporn Science High School Loi. Today we would like to present our project entitled The Development of Material for Entrapping Ammonia in Water by Polymer for Bacteria Cellulose Combined with Zeolite. Let's start with a background. Currently, a significant factor affecting the well-being of ornamental fish is water used for their care. It must be consistently managed and controlled. In fish tank, there contains ammonia produced by fish excretory system. The concentration of ammonia in fish tank should not exceed 0.01, 0.02 to 1 ppm. One method of removing ammonia in water is to use substances with absorption properties, such as zeolite, which is such a mineral that has these properties. It can undergo a process of absorption and release as small organic molecules and is reusable. Bacterial cellulose possesses a three-dimensional lattice structure, allowing it to hold a large amount of water with high purities. Therefore, we intend to form a cellulose from bacteria into a film in combination with zeolite mineral powder. This will be tested to assess its effectiveness in absorption ammonia in water. The goal is to further develop this concept for use in densely ornamental fish tank. The objective is to study the development of material for entrapping ammonia in water by polymer from bacterial cellulose combined with zeolite.
Let's go to the next point, methodology. First, produce cellulose from the bacterium Acetobacter solanum by using the coconut water. Boil the cellulose sheet with sodium hydroxide solution and wash the cellulose sheet with a water until the pH equal to 7 and break until dry. Next, Next, produce cellulose from the produce cellulose powder by guiding it and then mixing it with sodium arginate, glycerol, and zeolite. Continually stirring for 3 hours. Then the mixture is poured into a plate and break until dry. Afterward, the material is tested for its efficiency absorbing ammonia and reusability by using the Finette method. And bring the solution to measure the absorbance with a UV spectrometer. Furthermore, design innovation to use in the fish tank. The picture showed the amount of ammonia remaining. From the experiment, it was found that cellulose sheet in one the highest efficiency in ammonia absorption, which occurred at a duration of 6 hours, resulting in 26.8% removal. Conversely, the lowest ammonia absorption efficiency was observed with cellulose sheet from sodium carboxyl methyl cellulose mixed with 2.5 grams of zeolite, which occurred at a duration of 0.5 hours, resulting in 10.1% removal. And the picture shows the recycle efficiency chart. Regarding the study of reusability over 5 cycles, statistical analysis using one-way ANOVA was employed to compare the statistical significance of the efficiency in each cycle. The research showed that there was no significant difference statistically, alpha equal to 0.01 or at 99% confidence level. And here is a simple video of using ammonia absorption sheet together with innovation.
Thank you for your kind attention. Do you have any question?
Thank you for your excellent presentation. Next, we would like to introduce our commentator to give advice or comment.
Hello. Okay, all right, Scott. Okay, good morning, and thank you very much for your nice presentation. May I ask the principle behind the concept of using the cellulose and zeolite? I understand that zeolite has the ammonia absorbance capability. What about cellulose? Why do you have to mix them together in order to trap the ammonia?
Because cellulose have a three-dimensional elastic structure and it's made for a bacteria, then it will be a nanocellulose, and it can absorption. It can hold a large amount of water. Yes, and it's different from different cellulose because it have a high purity. It's not consist of lignin or pectin. Yes.
Thank you for the presentation. I would like to ask, how long did it take for you to, from the start to finish, to make one working product?
Since I was starting this concept, or I start the experiment. Since I start this project, it's about maybe 2 years, because I start do this project since I am grade 11, and I do all of the journey to make this. I think the longest step is to test its efficiency. Yes. This step is the longest, and and this is the longest step too. It used time maybe for 3 months.
Which takes the longest time to make? Is it the waiting for the cellulose to be made by the bacteria? Or?
Yes, we use a coconut water to make a bacterial cellulose sheet. It waits for 15 days, and we not only do it one time but many times, so it take a long time.
I think that's very interesting that you use coconut and bacteria to make it. I think it would be great if you can market it more, like put coconut on there. We use sustainable materials or something like that. That would be very interesting as well.
In Chiang Khan, they have coconut street, and they don't use the coconut water. And
Prepare the coconut from the waste of the coconut?
yes,
Oh, okay. And what is the status of your experiment right now? So you have the cellulose with the zeolite, right? And you put into the water as you show in the video. So what is exactly the status of that experiment?
We prepare to looking forward to make it for product to use in shop, and everyone can buy it, especially ornamental fish on the tank.
Have you tried yet with the ornamental fish tank?
We have a prototype, but now we find what design is the best for fish, because
Okay.
we look at the best design. this design maybe maybe is must wait a minute. Okay, never mind. This design, you will see that if we are spin right,
Yes, okay.
is maybe it must make a fish straight, you know.
Oh, I see. So can you just put them into water without aerations?
Yes, maybe we have to put a projector,
Which,
protector.
okay, all right.
Will just out of curiosity, will the fish eat the cellulose made from the coconut? If you put it in a fish tank and it's not protected,
I think it's not a good. I think the fish doesn't like it.
have you tried put it in a tank with a fish though? Have you? Mm,
I ever try, but you know, this design is not fit for a fish. But this is you know, we can develop it for outer design.
So how big of the sheet of the cellulose and zeolite? How big is that? The one that you put in the tank? Yeah, what is the size?
1, 2, 3, 4, 5, 5 sheets.
5 sheet, but what the size? The width?
Yes, 3 centimeter, 3 centimeter multiplied by 6 centimeter,
Oh, 3 by 6.
3 by 3.
Oh, it's a big one, so well, I'm not sure the fish may not eat it, but it's just like a. Okay, so what can we use other material besides the coconut?
We use Acetobacter because it not easy to find it in market, and is give us a pure cellulose so fast. Yes. It's
You can try other materials, other ways that yet can be used, and maybe to optimize what types of material may be fit or for the production of cellulose from this bacteria. Yeah, just maybe another comments.
Thank you. Thank you for your kind attention again.
Thank you, the feedback commentator.
We would like to invite our commentators, Associate Professor Dr. Vilaylakshmi Siriphorn Nurasin, onto the stage to give the certificate of presentations.
The next presenter from Oyama Kosen, which the project title Isolation, Identification and Characterization of Peony Bacteria from Japanese Dairy Products. Please come on to the stage.
Test.
Hello everyone, I'm Futaba Naka from National Institute of Technology, Oyama College. I'm very pleased to have this opportunity to speak to you. Today, I'd like to talk about my study of Propionibacterium. My presentation consists of 9 parts. First of all, I'd like to start by talking about Propionibacterium. Propionibacterium is gram-positive, non-model, non-spore-forming, and anaerobic to air-tolerant bacteria. Furthermore, daily Propionibacteria have the production of propionic acid and vitamin B12, immunomodulatory, anti-inflammatory, and bifidogenic growth stimulatory effects. They are found in dairy products such as cheese and raw milk, as well as in silage, and mainly used in the food industry, such as cheese ripening and the production of propionic acid and vitamin B12. But they are not almost found and discovered in Japan products at this time. The present study aimed to isolate and identify Propionibacteria from dairy products like cheese, butter, and raw milk that produced in Japan, and to qualify their characteristics. Next slide, I'd like to show you the method of our research. 5 microliters of the diluted samples were applied to YEL agar medium and incubated anaerobically at 30 degrees Celsius for 1 week. Each tentative colony was picked up and streaked on YED agar medium and incubated anaerobically at 30 degrees Celsius for 1 week to check the cell separation and acid productivity by catalase test and the dissolution of calcium carbonate. The formation of oxygen bubbles on the colony was averted as catalase positive, and the formation of clear zone around the colony was averted as positive. Finally, isolated strains were incubated in YED medium and cultivated anaerobically at 30 degrees Celsius for 3 to 5 days. The supernatant was obtained by centrifugation of culture broth and organic acid production was averted by using HPLC. Propionic acid producing strains were identified by 16S-rDNA sequencing. Each strain was precultured by centrifugation. Bacterial DNA was extracted using nuclear spin tissue. DNA was amplified by PCR using the bacterial 16S-rDNA PCR kit. Finally, the obtained DNA sequences were compared with those in the database by using BLAST. In the next slide, I'd like to show you the data from our study. In this study, 6 strains were selected by their characteristics as catalase test and acid productivity. In particular, strains number 14, 18, and 36 produced high amounts of propionic acid and acetic acid, with organic acid to glucose used. This table indicates the identification and characterization of isolated strains. In this study, we were able to isolate and to identify some propionibacteria from several Japanese dairy products, such as Emmental type cheese, Gruyere type cheese, and blue cheese, and uncultured bacterium producing propionic acid from raw goat milk. 16S rDNA sequence analysis revealed that the 5 strains were most closely related to propionibacterium. Strains number 37 was most closely related to the uncultured bacterium that had not been successfully cultured and related to and reported only as environmental DNA. In conclusion, we have successfully isolated and identified 6 strains of propionic acid producing bacteria from 75 dairy products. In particular, the isolation of propionibacterium from blue cheese and the isolation of uncultured propionic acid producing bacteria from raw goat milk are new discoveries in this study. We will investigate the functionality of these isolated bacteria, such as the potential of bifidogenic growth stimulator, and examine the application to industry in the future. That's all. Thank you for your time.
Yes. Thank you for your excellent presentations.
Next, invite commentator to give advice or comment about this project.
Thank you for your nice presentation. So you mentioned that this bacteria was not found in Japan products. Could you sort of clarify this sentence?
Sorry, could you say that again?
Yes, in one of your slides, you said that this bacteria, the propionic acid bacteria, was not found in Japan products. Yes.
I mean, some dairy products like
So your idea is try to use this bacteria in the Japanese products in the future, is that
yes.
right? Okay. All right. So in your isolation slide, I think it's the very first. Could you go back to? Yeah. Okay. This slide. So you try to isolate the bacteria from 3 tri-up sample, right? What kind of samples that you use?
75 dairy products, like
Dairy products. Okay.
yeah.
So there is milk as
Yeah,
the
raw milk and
Raw-
cheese and
Oh.
butter.
Okay. So from this type of the screening, can we tell that these are the propionic bacteria? How can we identify that?
So, mm,
Like from the first step, you mentioned about the catalase test. Yes, sir. So what is the principle of using catalase test to check for the propionic bacteria?
yeah,
Yes.
to check the acid productivity,
Ah,
like
okay.
formation and reaction is here, and this mean like acid productivity, and
Uh-huh.
this shows acid
The bubble.
Yeah,
Yes, the bubble.
like
Okay. So
oxygen.
right, the bubble of oxygen. So actually the catalase test should be positive
Yeah.
for this type of bacteria,
Yes.
right? And
Yes.
for the acid, you can use the zone, right? The creosote over here. Yeah, that could like dissolve the calcium carbonate in the samples.
Yeah.
Right.
Same as like this reaction.
Okay. And then how can you identify whether this is propionic bacteria?
After this after this test, and we picked up like this colony
Okay.
from the medium.
For the medium. Okay. And I saw your HPLC experiment, right? You have HPLC experiment.
Yes.
Do you have that? Yes. Could you next slide, please? No. There's the graph that you show the presence of the acetic acid and propionic
Yes.
propionic acid.
Organic acids.
Right. So you check that with the HPLC.
HPLC. But our laboratory don't have the these machines, so we send to the company.
Oh, okay. So okay. So you identify them using the 16S.
Yeah.
Yeah. So you get the final. You get like a 6 strain, right? 6
Yeah.
strain. Go to the blast table. Do you have blast table? Yeah. So are they the same, or because in the crowset pyrogenic relative, I saw like the same strain of FAM 143. Yeah. Are they? Could they be the similar strain or the same strain?
Yes. Here, 100% or 99%. We would like to search about this uncultured bacterium. Yeah. This is only reported environmental DNA, so we are research about this bacteria more and more.
Characterize more on this like a
Yes.
4 strain that you have over here.
Yes, and we also want to create cheese with isolated strains and bacteria. Yeah. Thank you.
Thank you for the feedback, commentator. We would like to invite our commentator, Dr. Hatchapa Deshakaisaya, onto the stage to give the certificate of presentation.
Please clap your hand.
The next presenter are from Princess Jalapon Science High School Buriram with the project title A Study of Optimal Ratio of Medicinal Plants Containing Alkali as Ingredient in Liquid Bedded Gel to inhibit the growth of Staphylococcus aureus, the cause of inflammatory. Please come onto the stage.
Hello everyone, my name is Navarashiram.
And I am Bingkop Sanchayat.
We're from Princess Jalapon Science High School Buriram. Our project is a study of optimal ratio of medicinal plant containing alkali as an ingredient of liquid banded gel to inhibit the growth of Staphylococcus aureus, the cause of inflammation. Using traditional wisdom about medicinal plants for healing and inflammation, we decide to dig deeper into this topic. And when we find out that infected wounds are often connected to Staphylococcus aureus bacteria, we decide to create a liquid banded gel with strong
Get the extract from the two Thai medicinal plant. We will in it differently. For the Prima herbaceae, we use 10 grams of herb washed ground and dye it, and then soak it in hexane and methanol for a week. On the other hand, we use 20 grams of Mithec pinnata powder in solid extractor with ethanol-based liquid solvent. After filtering, the substance will evaporate to get a dry extract. And the next, show the preliminary phytochemical test. To detect the alkaloid in Premna herbaceae, we use Wigner reagent. The color will change to yellow, brown, or yellow. And to identify the glyphosate in Vistec pinnata, we use chloroform with hydrochloric acid. The color will change to reddish brown. And this slide shows how well does the extract inhibit SOS. We use the broth dilution test. First, we cool the bacteria in nutrient agar and then transfer the colony to the nutrient broth for activation. And then we measure the optical density by use spectrophotometry at 0 hour at 600 nanometer wavelength. And then we bring the test tube with nutrient broth and extract at various concentration as 150, 25, 12.5, 6.25, and as amoxicillin as a control, and cause a dose of bacteria each tube. After 24 hours in at 37 Celsius degrees, we check how well does the extract inhibitory the SOS by we look at the turbidity and to get the gel from the tamarind seed. We started by washing the seed and heated it and peel off the seed coat. And then the seed will turn into the fine powder, and entering all the particles were the same size. So shifting. To get the liquid gel with the extract, we use the tamarind seed gum powder dissolving in the saline and then adding the extract based on MAC value. And the last method, we check how well does the liquid gel extract the SOS by we use the morphylation test. We set up the test tube with NB and a gel each, and then having NB with gel in a negative control and having a gel with amoxicillin in a positive control. After incubating at 37 Celsius degrees 24 hours, we check how does the liquid gel inhibit SOS by looking at the turbidity. And for the results, this table shows the preliminary phytochemical test. We found alkaloid group in the Premna herbaceae and we found the glyphosate group and Vitex pinnata. And this graph shows the efficacy of Premna herbaceae extract work against SOS. At 100 microgram per milliliter, can against SOS at 47.2%. And Vitex pinnata extract can work against SOS. You can see from the elevation of graph are almost the same. And the last results show liquid gel efficacy through the SOS. The liquid gel can work against SOS at 50%.
To sum it up, we successfully created a liquid gel using alkaloid and glyphosate extract from tamarind seeds. This liquid gel holds great promise for various medical applications, especially in the realm of wound healing and other therapeutic purpose. However, it's important to note that future studies are needed to thoroughly assess its effectiveness and safety before considering it for potential clinical use.
Thank you for your excellent presentations. Next, I invite commentators to give advice and comment about this project.
We use a concentration of positive control list. We use 5 microgram per milliliter. The graph in each concentration are almost the same, the negative control. We conclude the Vitacrinata extract can be able to against areas.
Yeah. So exactly what is the positive control and what is the negative control in your understanding? What is positive control? What
Positive-
did you put in your tube that you said is this positive control?
In
What
positive
should be?
control, we
Yeah.
put,
Yeah.
we put gel, pure gel and amoxicillin and nutrient broth.
Is that right? Actually, to me, the positive control means that the Staph aureus should grow normally in the nutrient blood. So it should have only these two things, bacteria and the nutrient blood, that they can grow. So you make sure that when you see the turbidity, the bacteria could grow normally. There is no problem with your bacteria. Then in the negative control, normally you should put just a little nutrient broth into that. Is that right? To my understanding. So nothing should grow in there. Yes. So the turbidity in your graph should be very low in theory. Okay. But in your experiment, in your treatment, in the maybe treatment 1, you have NB, you have bacteria, and you can put some liquid bandage gel in that. That is the experimental treatment that you should see the difference. Okay, but in your graph, you see, the level of all the turbidities are the same. So I think we cannot conclude anything from this experiment because the negative control is not right. It should be very low. Yeah, and the positive should be very high. And in your treatment, it depends what that would affect the bacteria. Okay, this is my comment. Okay, thank you.
Thank you for the feedback, commentators. We would like to invite our commentators, Associate Professor Dr. Vilailak Siriphorn Nurasin, onto the stage to give the certificate or presentation. Please clap your hand.
The next presenter are from Oita Prefectural Maizuru High School with the project title The Antimicrobial Effect of Kodapa Owata. Please come onto the stage.
SP. The bacterial colonies were confirmed using the standard medium, but not bacterial colonies were confirmed Catarrhobacter agar. For this reason, reason, this year's research question was, does Caterpillar has an antimicrobial effect? And we conducted research based on the hypothesis that Caterpillar is effective against fungi and the bacteria. Next, we will discuss the materials and the methods of the experiment. This is about how to make an extract and the agar medium. The water extract is completed by adding water to Caterpillar, boiling it for 15 minutes and filtering it. The acetone extract was completed by adding 60% acetone to Caterpillar and extracting at room temperature for 3 days, followed by distillation under reduced pressure. Add DMSO to the distilled solder and completed. The hexane extract and the hexane water extract were extracted in the same way was the acetone extract, followed the distillation under reduced pressure. After that, water and hexane were added to the distillation solid and the separatory. Finally, was used to separate the hexane cyber part and the water cyber part. The hexane cyber part is distillated over under reduced pressure and completed in the MSO. The water cyber part is filtering and completed. Next, in the agar medium, five types of agar medium were used. The leads of water and extract is as follows.
First, let us discuss the effect on twig phyton bloom. This is a twig phyton bloom's condition. There are significant differences between the standard medium and acetone agar, and standard medium, hexane water agar and hexane agar. Let's look at the discussion. From the result of the experiment, we consider that the lipophilic component in the acetone extract and hexane extract, but not in the water extract, show a growth inhibitory effect on twig phyton bloom. Next, let's look at the effect on Caterpillar. Next, let's look at the effect on Caterpillar butter on bacteria. First, let's look at the Staphylococcus aureus. This is Staphylococcus aureus condition. From the graph in those figures, we have found significant differences between standard medium and water agar, and standard medium and acetone agar, and standard medium and hexane water agar, and hexane water agar and hexane agar. Next, we will look about Elizabeth Kingia. This is Elizabeth Kingia's condition. From these graphs, we have found significant differences between standard medium and acetone agar, and standard medium and hexane water agar, and hexane water agar and hexane agar. Let's look at the discussion. From these results, we believe that the hydrophilic component in the Caterpillar water extract, the acetone extract, and the hexane water extract are present in Caterpillar water. Let's look at the conclusions. Through this study, we have confirmed that Caterpillar water is effective in inhibiting the growth of twig phyton bloom and two types of bacteria. We believe that the effect on twig phyton bloom may be due to the presence of lipophilic components similar to those found in antifungal drugs such as azole and purines in the constituents of Caterpillar water. On the other hand, against the two types of bacteria, we believe that some of the components of Caterpillar bacteria are similar to the hydrophilic component found in antibiotics such as aminoglycosides and tetracyclines, which may be responsible for its effect on both gram-positive and gram-negative bacteria. It is also possible that the growth inhibition effect is due to an unknown component other than the reported. This is the future work. Thanks and references as follows. Thank you very much for your kind attention.
Thank you for your excellent presentations. Next, we would like to ask our commentator to give comments about this project.
Thank you for your presentation. So there are two types of components, right? The lipophilic and the lipophilic, and what is that in water? Hydrophilic. Oh, okay. So why these two components have different functions in terms of the antimicrobials against fungi and the bacteria?
Sorry, once again, please.
Okay. So what are the functions of the lipophilic components? Okay, and the hydrophilic components against the fungi or bacteria that you use in this experiment? Yeah.
Okay.
What mechanisms to explain that? Yeah, how? Yes.
We use four type of extract. This water extract, acetone extract, hexane extract, hexane water extract. So those extract has so water agar has only hydrophilic component.
Okay, all right. Okay.
Acetone agar is both hydrophilic component and lipophilic component.
Okay.
Hexane water agar is only hydrophilic component, and hexane extract have only lipophilic components. So we think that from the result, we think that the efficient extract has component. This is effective for to fight or bacteria.
In terms of the structure of the microorganisms, like the fungi have some structure that would be affected by the components. Yeah, I mean in terms of structure of the fungi and the bacteria are different. Yeah.
Yeah, I think so.
Okay, so how would you identify these components, those hydrophilic or lipophilic components in the extract?
For example.
Yeah, how will you identify them in your plant? How could you identify them? Yeah.
For example, lipophilic component and hydrophilic.
Yeah.
We think the lipophilic component, for example, iridoyl glucoside. This is iridoyl glucoside. 4-hydroxyl benzoic acid is in Caterpillar butter has lipophilic component about this. So on the other hand, the hydrophilic Caterpillar's hydrophilic component, for example, this is flavonoid in Caterpillar is flavonoid in Caterpillar. This is hydrophilic component.
HPLC or something to identify them, to say, okay, this is azo or this is this component in terms of identification.
Yeah.
How can you tell which type of the chemicals? Yeah. Okay.
This information is all of from differences.
Okay.
So differences right about this. So this component has lipophilic or hydrophilic character. So this is the actual drinking tapa water. So this non-diuretic effect is often used for drinking. So, but this antimicrobial effect is so Catalpa and it so Catalpa says that has an antimicrobial associated with inflammation, so antifungal effect. But this is no science, no scientific paper, not be effective. So I can say that Catalpa has antimicrobial effect, but from this experiment, wishing Catalpa has antimicrobial effect and is effective on drinking our body.
Thank you for the feedback, commentator. We would like to invite our commentator, Dr. Hatapak Deshakaisia, onto the stage to give the certificate of presentation.
Thank you, the presenter and thank you, the commentator and presenter.
The next presenter are from Sapporo Kaisei Secondary School, and their project title is Plant Derived Sunscreen. Please come to the stage.
about our abstract of project. So sunscreen can protect our skin from UV rays. However, they sometimes contain ingredients that are harmful to the environment. For example, UV absorbing ingredients such as oxybenzone and oxynoxide can negatively impact on the marine life and and coral reefs by reaching into the ocean. Therefore, we thought that we want to solve this problem and we want to create an effective sunscreen without the use of UV absorbers. And as an alternative, we came up with the idea to use the plant pigments and so such as chlorophyll and cyanin, lycopene and carotene. So they have the ability to absorb UV rays, and they are not harmful to the environment.
The sunscreen element is water, jojoba oil, and the UV absorber and beeswax. So we changed the UV absorber to the plant pigment. And we have two objects. First, we want to find the pigment at the place of UV absorber. We think chlorophyll and anthocyanin absorb more UV. And the second is, we want to make the stable softness of sunscreen. We think that the more work, the more stable. We try to find the pigment that absorb UV. We crushed the vegetable in a mortar and made 3 liquids, such as the pure water, hot water, and ethanol, because in the previous research, chlorophyll dissolved in ethanol and anthocyanin dissolved in water. And we used the UV checker to measure the absorbed UV. It can measure SPF and PA, but it can measure how much UV was transmitted. The result is this graph. For this graph, the red perilla contains element of chlorophyll and anthocyanin, so we think that red perilla is the most of the UV absorbed. Next, we try to make stable cream. The elements of cream are water, oil, and beeswax, so we change the ratio of them and find out the best ratio. And also, we define that stable cream is kept creamy for 1 week, don't separate. And in this method, we measure how much water separates them. And as a result, the amount of wax is increased, the harder the cream becomes, and the more stable it becomes without water separating them. This is a graph of separate water from cream. So we think the 1.5 cream is the best.
In conclusion, we found that chlorophyll and anthocyanins are able to cut almost all UVs. So chlorophyll is fat soluble and thus easily dissolves in ethanol and not water. And anthocyanins, on the other hand, are water soluble and therefore dissolve well in water. Taking advantage of these two different characteristics, 2 different sunscreens were created. Both are considered suitable as sunscreens because the green color can remove redness from the skin and the purple color can increase the transparency of the skin. And in future experiments, we would like to conduct training on pigment formulation and effectiveness as a sunscreen. And we would also like to compare the effectiveness between 2 sunscreens and we want to put this idea into practice. Yes. These are references. Thank you.
Thank you for your excellent presentation. Next, I invite our commentator to give advice or comment about this project.
Thank you for your nice presentation, and the idea is very interesting. And what criteria that you select the type of vegetable for the using as the sunscreen or UV absorber?
I researched about the pigment, and for example, isocyanine like the melanin
Oh,
for
okay.
the human cell. So we think about the alternative to isocyanine is good.
Or anthocyanin. Okay.
yes.
So you select based on the type of the pigments of all the vegetable, and you found that chlorophyll and anthocyanin are the best. Yeah. So why chlorophyll and anthocyanin are the best UV absorber? That depends on the properties of the structure of these pigments, or yeah.
I researched about it, and this pigment protect the plant vegetable cell, and our melanin is protect our cell. So I think that the alternative is
Our skin was protected by melanin, but as a sunscreen, melanin is a little bit harmful and not suitable for sunscreen. So we researched about the as an alternative anthocyanin and chlorophyll. Yeah.
Is that okay to mix them? I mean, in terms you can mix like a chlorophyll and anthocyanin together. Have you tried that yet? Oh, is that working? Is that better than just only one type of pigment? Oh, okay. That's interesting.
I agree. Very interesting project and very bright potential, I think. So with your method of testing the UV absorption, can you? Yes. So you're checking how much UV is being absorbed and not going through the surface of something. Can you explain a little bit more how you measure the UV?
UV light max is 1.2, so we use the 1.2 light, and the red perilla absorb the 1.2, so almost UV is cut.
Okay. And what do you compare it with? Do you compare it with commercial sunscreen, or do you, what is your control? What is the control for this experiment? What do you compare it with?
We don't compare the commercial sunscreen.
Oh, you use the commercial sunscreen, or
No,
oh, no commercial.
not
Okay.
yet.
Yeah, it would be nice to see as well, like how much commercial sunscreen can block the UV. For example, SPF 20, 30, 40, 10, 50, what so and so on, right?
I want to research about the SPF or PA, but our school don't have the
Understand.
okay, so
Actually, there are several types of UV, right? UVA, UVB, UVC. So in your experiment, 1.2 is equal to what?
UVB.
UVB. That's the most dangerous one. Oh, okay. Maybe you can try others. Yes. So in these experiments, you just apply. So you make the sunscreen, right,
Yes.
from the pigments,
Yes.
and you apply that on how do you apply that? Could you go to the next slide, please? Yes, how you did experiment? Yes, this one. What's that sheet? Yeah, what did you put it on? The white? Yeah. You see the white piece? Yeah. How did you apply that? And how you did it? How did you do the experiment?
She has a real sunscreen. Yeah. And we first we put the sunscreen into plastic, plastic, and push it and make it flat. Yeah.
Thank you for the feedback, commentators. We would like to invite our commentators, Associate Professor Dr. Vilailak Siriporn Dulasin, onto the stage to give the certificate of presentation. Thank you, the commentators and presenters.
The next presenter are from Chawat Vidyakan School with the project title Study on Soil Quality Affecting Percentage of Latex Strain RRIM 600 in Chawat Nakhon Si Thammarat. Now, break 15 minutes. Sorry, 5 minutes.
Thanathorn Thongsang.
My name is Chanatip Demnun.
We are students in grade 10 from Shawat Withikan School, Nakhon Si Thammarat Province. Today we are going to present our project that named Study on Soil Quality Affecting Percentage of Latex Strain RRAM 600 in Shawat, Nakhon Si Thammarat. Well, I'm going to start with the introduction. Rubber, mostly known as heavier bacilliosis, that play an economical role in agricultural products in the south of Thailand, especially in Shawat, Nakhon Si Thammarat. The rubber trees at latex strain RRAM 600 is a famous plant for this area because they can adapt to almost any environment and yield well. Shawat area had a different geography. Therefore, we are interested in studying the soil quality that affects the percentage of latex strain RRAM 600 in two areas, the Kwan Nong Hong and Khao Pratong sub-district. Furthermore, we choose 10 years of latex for this study. And next topic is objective. Aim of this study to investigate the quality of soil which affects the percentage of latex strain RRAM 600. So we will be moving to the next topic is methodology.
There are three parts of this study. Part 1, we study the soil texture by using a field soil inspection manual. Part 2, we study the quality of soil in different story. Part 2, we study the quality of soil nutrient, nitrogen, phosphorus, potassium, and pH value. We decide by 6 steps. And the last one, we collect the data in 6 months during January to June 2022. And the next part will go to the result and discussion. The results show the soil quality of Khao Pratong is in type of sandy loam, while loam is found in Kwan Nong Hong. Furthermore, rubber tree globe well in sandy loam that is found in Khao Pratong. Next slide shows pH of two areas between Kwan Nong Hong and Khao Pratong. Blue bar graph shows pH of Khao Pratong 7.33 is considered neutral, and the red bar graph shows pH of Kwan Nong Hong 6.67 is slightly acidic.
Testing the quality of soil nutrients NPK based on a comparative analysis between Khao Pratong and Kwan Nong Hong. Blue bar graph show the key nutrients in Khao Pratong, which were found to have values of 0.47, 43.33, and 66.67 milligram per liter, respectively. Furthermore, the red one show NPK in Kwan Nong Hong are 0.1, 23.33, and 23.33 milligram per liter. The primary nutrient nitrogen, potassium, and phosphorus in the Kwan Nong Hong are greater than in the Khao Pratong. Moreover, the NPK in Khao Pratong, which were in rank of standard nutrients. This study showed that the collecting data in 6 months, January to June in 2022, blue line is Khao Pratong and red line is Kwan Nong Hong. The percentage of latex indicates that the value ranks 31 to 36 in two areas. The Kwan Nong Hong area is less valuable than the percentage of Kwan Nong Hong area.
Conclusion: the quality of soil in different of Shawat impact the percentage of latex strain RRAM 600. This research indicates that the percentage of latex in Khao Pratong is greater than Kwan Nong Hong. Moreover, the soil texture of Khao Pratong is in type of sandy loam. pH is considered neutral. The primary nutrient NPK ranks standard. Nutrient concentration that is benefit and suitable for rubber plantation.
Okay, that brings the presentation to the end. Thank you for your attention.
Thank you for your excellent presentations. Next, I invite commentator to give advice comments about this project.
Okay, Thank you for your presentation. So you measure just really the amount or the quality, you said amount of quality of the latex. So what did you measure from the latex? Yeah, how did you measure that the soil would affect the rubber?
Yes, the soil is so important for rubber, but because the rubber can grow up well in the sandy loam.
Sandy loam, okay.
Yes, ซึ่ง sandy loam is in found in เขาพระทอง Yes, and loam is found in ควนหนองหง
Okay, so the type of soil,
Yes.
that's the first factor. And then the type of soil, would they affect NPK? Just really the type of soil or other factors in the soil? Why N is so low in both type of soils? Yes, N is very low. And for the P and K, okay, you said the Khao Phathong is higher than the other one. That could be the type of soil, right? The sandy loam.
Yes.
Yeah. So what about the N? Why the N are so low?
N is found in range of standard in 0.3 to 0.5.
Okay.
Yes.
So set in the standard level.
Yes.
Okay. So you measure the rubbers or latex. You measure the amount of latex that you get, right?
Yes.
The percentage. What do you mean by percentage of 33 to 36?
33.
Yeah. Yes. Okay. What is this? What is the percentage of latex? What are they? What do they mean?
Collecting data in
yes.
6 months,
Yeah.
January to June.
Yes. What does number means? What is 33%, 32%? What is it?
In
How did you measure it? The amount of latex that you get?
Yes.
Is that right? Yeah. What is that? Yeah. What is
percentage?
Yes.
In
Yes.
this month.
Right. In this graph. Yes.
Number of rubber.
How did you calculate this number on? How did you do the experiment? Did you collect the latex from each tree? That would affect not just really the property of the soil at the first time that you collect. For example, you collect the soil in January, but after that you never check it, so you don't know that that might be the variation of NPK during this month. Have you done that, or you just correct the soil just in one time?
Okay.
Do you understand? How many times that you collect the soil sample to measure the NPK?
One time.
One time.
Yes.
In which month?
In yes.
In which month? When? Yes, when? Jan, Feb, or in which month that you collect the soil sample? ไปเอาดินมาเมื่อไหร่คะไปเอาดินเดือนไหน
January ครับ
January คำถามก็คือเราแค่ collect sample แค่เดือนแรกเนาะแต่หลังจากนั้น 6 เดือนจะไม่รู้ว่ามันจะมี variation ของ soil property ตรงนั้นหรือเปล่าซึ่งเราไม่รู้ว่าความแตกต่างที่เกิดขึ้นตรงนี้เนี่ยมันเป็นจาก soil property จากเดือนแรกเท่านั้นหรือเปล่าคือถ้าจะให้ดีเนี่ยถ้าเราสามารถเช็คได้มากกว่า1 เดือนระหว่าง along the way จนถึง 6 months แล้วเปรียบเทียบ property of soil แล้วก็ latex ที่เราได้อันนั้นมันจะเป็นข้อมูลที่ค่อนข้างชัดเจนขึ้นนะคะขอบคุณค่ะ
So yeah, building up on that, what happened in May? Why does it go up, or April even?
Because rubber is this is in Jan to May, and relations restoration in April.
Restoration. What
is ฟื้นฟู grow up ก็คือ
so
ในช่วง January to May นี่ครับยางมันจะผลัดใบซึ่งจะมีการสังเคราะห์แสงได้น้อยกว่าปกติ
so in April is a peak
Yes.
peak time for harvesting rubber.
Yes.
Okay. Yeah. So it would be good for you to explain, put some information for the people who are not in the field of rubber making, for us to understand like why is there a peak, or do you measure it? How many trees do you measure it from, for example? Yeah, that's about it.
Thank you. Okay, thank you.
Thank you for the feedback, commentators.
We would like to invite our commentator, Dr. Hattapak Desha Kaisia, onto the stage to give the certificate of presentation. The next presenter are from Princess Jalapon Science High School, Mukdahan, and their project title is Innovative of Plastic Nanofiber from PVA for Plant Tissue Culture. Please come to the stage.
And my name is Nishara Tilakanapat.
And my name is Jiranan Salapat.
We're going to be presenting about our project, the Innovative of Plastic Nanofiber from Polyvinyl Alcohol for Plant Tissue Culture. Let's get started. So recently, tissue culture has been introduced and get to know more by lots of people as an alternative ways to grow lots of plants. To put it more simple, tissue culture is like putting in new plants in an artificial medium under strictly controlled and germ-free conditions. Now let's take a look at what's its causes and benefits here. Start up strong with disease-free propagation. Since it has been kept under strictly controlled and germ-free condition, it is safe to say that these plants are completely uncontaminated. And secondly, repeat multiplication with such a short period of time and limited space as well. Lastly, since it has been observed and contained in such conditions, I can say that weather and season are not considering a necessities anymore. For disadvantages, firstly, just like any types of cultivation, plants also need its own food resources. In this case, food or medium has to be changed once every 5 to 6 months. Which then leads to another problem here, that medium can cause a thorns, which can be troublesome in the long-term propagation because it's going to cost a lot of money. And lastly, we also need a lot of manpower in order to take care of the plants, and it only going to add up extra expenses here. That's when we came across polyvinyl alcohol to be using instead of any regular agar medium, which has an outstanding characteristic including durability, top-notch water absorption, and well suited for synthesizing into fiber shape, and lastly, elasticity. As a result, these types of fiber is the best one yet to reduce, minimizing the risk of contamination and leading to cost saving, and of course, it is good for using as a medium. For the objective, firstly, investigating the optimal concentration of polyvinyl alcohol for plant tissue culture. Secondly, examining the water absorption characteristic. And lastly, identifying the most effective concentration of polyvinyl alcohol to be using in a plant tissue application. We're going to start with experimental process. Start with PVA solution preparation. We're going to be prepare PVA solution in a distilled water on a hot plate by using a magnetic stirring bar and continuously stirring for 2 to 3 hours. And then with the fabrication of plastic nanofiber, we're going to be using transferred solution into a syringe, attach the needle, cut off the tip for a smooth flow, and then attach the aluminum foil to the rotating collector. Then place it away from the needle 25 centimeters with a pumping rate of 2.22 meter per minute and a pumping here with a 2.8 milliliter per hour. Then allow this collector to run for 6 hours straight with 25 kilowatts high voltage. And we're going to be examining the attributes of plastic nanofiber sheets. Start with structural analysis. We're going to be analyze the structure of the nanofiber sheets produced to SEM. As you can see here, this is a plastic nanofiber sheets. And we're going to be talking about the water absorption assessment. Prepare nanoplastic fiber sheets at various concentration, then calculate the water absorption efficiency from fiber mass post-absorption minus fiber mass pre-absorption, then divided by fiber mass post-absorption all over again. And prepare the plastic nanofiber sheets, then sterilize the sample by soaking in 25% of sodium chloride, then proceed to rinse it thrice. As you can see here, it was white before, but now it's completely transparent. And then introduce the sheets into a medium contained with a bottle of Drosera spatulata, which has undergone 1 month of propagation already.
As a result, different concentration result in varying consistency that will rather impact the structural and characteristic of plastic nanofiber. For experiment 1, revealed that plastic nanofiber form PVA 12% and 14% weight by volume concentration has polymer doublet and uneven fiber diameter. On the other hand, plastic nanofiber form 16% weight by volume concentration, give off the smoothest fiber and without polymer doublet. For this table, we discovered the plastic nanofiber from polyvinyl alcohol 16% weight by volume has the highest water absorption, followed by 14% and 12% with by volume concentration, since 16% concentration of PVA solution has the contain higher PVA and form the thickest fiber, so which result in the higher water absorption. It has confirmed by the measurements of diameter, quantity, and length of load-independent growth. The plastic nanofiber is suitable for plant tissue culture. As you can observe from this table, it shows the plastic nanofiber from PVA 16% with by volume had the highest growth rate. So we can conclusion the plastic nanofiber derived from polyvinyl alcohol with a concentration of 16% with by volume stand out as the most suitable choice for plant tissue culture. Thank you.
Thank you for your excellent presentation. Next, I invite commentator to give advice or comments about this project.
Okay, thank you for your presentation. So the idea is try to use the plastic nanofiber to replace the media, or
yes,
right?
Yes.
No media in the tissue culture bottle, or
Have
so
we
what the difference? Yes.
we used plastic nanofiber instead of agar,
So
but
Mm-hmm.
put the medium
So the-
and plastic nanofiber will absorb the medium for plant tissue culture.
so why is better than just read the medium?
It's going to be needing less medium in, yeah, so usually it would take like 20 grams, but after using this sheets, nanofiber sheets, it's going to be reduced into like 16 grams. It can like lead to like cost saving, which can be effective in the long run.
Oh, okay. So in your table, the last table, do you have the control of the normal agar compared with this experiment? I mean normal, in the normal condition of the agar mediums, and use the PVA plastic as the treatment. And do you see the difference? Is that better?
For after we're using the sheets, it's like we can see like different in the plant growth, that this one is like better than the normal agar that we use.
Oh, so you have that control.
Yeah, but we just didn't put a table here.
Oh yeah, okay, you should put that here too, actually. And actually you should have like the average. I would say if you have plant number like 1, 2, 3, just plant diameter, you average them. Yeah, that's my recommendation, to average them. You just have one number for each treatment. Okay. Let's see, and you mentioned that the 16% is the best. Do you have the reference, the percentage that you use? Can we use higher than 16 and you might get a better results?
If it's higher than 16, the machine, the electrospinning technique won't be able to work because it's too thick. Yeah, we have to be able to make the liquid into a fiber shape. So if it's too thick, then it cannot be making into the fiber and it's not be able to use. So that's why 16% is the best for now. Oh, I mean, still labor intensive, but still, I mean, it's going to be less frequent to change it, because usually it would take like 5 to 6 months to change per round. But after using the sheets, is required to change like 8 to 9 months. So it's like reduce the change of medium frequency. So yeah, still. Machine costs a lot, but yeah, after that, if we like bought the machine, and I think still it's like really cost saving too. It is very good, biodegradable. It is biodegradable. It's okay.
Point to look at the degradation of this plastic, whether it's dissolved in the agar and to the plant, directly to the plant. That is very challenge to to look at that, and also the cost of PVA. I think that the price is quite high for the PVA. Is that right? The PVA, the polyvinyl alcohol, I think it's quite expensive. Can we have other types of materials to make the plastic nanofiber instead of chemicals? Is that possible?
As for now, for our field, I do think the PVA is still cheaper than the normal agar that we use. Like we mix in the medium, so still it's like a cost saving.
Okay. Okay, and maybe it might be better to look at the degradable rate, like let's see, 1 month, 2 months, 3 months, and you measure the change of the plastic in the bottle, and then you can have some information to, for the next applications. That's what I think. Okay, thank you.
Thank you so much for your kind attention, and thank you for everyone.
Thank you for the feedback, commentator. We would like to invite our commentator, Associate Professor Dr. Vilaylak Sriphondulasin, onto the stage to give the certificate of presentation. The next presenter are from Seishin Kakuen High School and Junior High School. With the project Thermotolerant Yeast, please come onto the stage.
Hello everyone. We are very glad to be here today. Before our presentation, let us introduce ourselves. I am Miki Sayama and
Nananemoto.
We are from Seishin Gakuen. Our research team is thermotolerant yeast. And I think some of you think that what is yeast? Yeast is a fungi that reproduced by budding. And yeast is used making bread and making alcohol in industrial uses. our objective is finding the way to improve heat resistant ability of yeast because heat is important factor in industrial uses. So we and we hypothesize that yeast become to be able to grow at higher temperature by increasing the growth temperature step by step. So now we going to talk about our experiment. First, we extracting yeast from flowers blooming our school. First, we collect flowers and put in them in liquid medium and it is a kind of culture has high ability of fermentation, but our flower yeast is not the species. I'm going to talk about the experiment 1. Comparison of growth temperature of flower yeast and the baker's yeast. Method, we cultured baker's yeast and flower yeast at 40 degrees and 44 degrees and check the cultivation status. And second, we cultured baker's yeast and flower yeast at 40 degrees and 42 degrees and 43, 44, 45 degrees and cultured and check the cultivation status. This script shows the result of experiment 1.
This is the results of This is the results of experiment 1. We considered if no colonies appeared that yeast to be dead. In this way, flower yeast colonies appeared and yeast grew at 44 degrees. And at 45 degrees, no colonies appeared and the yeast died. Baker's yeast, on the other hand, grew at 42 degrees and died at 43 degrees. For these results, we considered flower yeast has better heat resistance than baker's yeast. In experiment 2, we investigated whether heat resistance could be improved by increasing the temperature in stages. The temperature was increased from 40 degrees by 1 degree every 2 days. This slide shows the results of flower yeast. Flower yeast grew at all temperatures up to 44 degrees. We haven't tested at 45 degrees yet. Baker's yeast also grew at all temperatures at up to 44 degrees. This is a comparison of the results of experiment 1 and experiment 2. In experiment 1, Baker's yeast only grew up to 42 degrees and died at 44 degrees, but in experiment 2, Baker's yeast could grow up to 44 degrees. From these results, it can be said that the heat resistance of Baker's yeast was increased by gradually raising the culture temperature. Then I'll explain the viewing. I would like to research how much flower yeast grows by gradually increase the culture temperature. Last year, we were not able to perform the fermentation test. Well, so we would like to conduct the fermentation test again. Heat resistance is also known to be given by dry stress. So we expected that Baker's yeast, which is so dry, so would be more heat resistant than flour yeast. But actually, flour yeast has more heat resistance. Therefore, we think that flour yeast may have higher heat resistance than Baker's yeast. And we would like to investigate it in the future. These are the references. Thank you for listening.
Thank you for your excellent presentation. Next, I invite our commentator to give advice or comment about this project.
Okay, thank you for your presentation. When you define thermotolerant, is there any like a specific temperature that you can use this thermotolerant? I mean the level of temperature that you can use to define them, like a 40, 41, 42, 45, something like that. So you have to heat it up until like a 45 and you can call thermotolerant. Do you understand my question?
I'd like to
Mhm.
do the experiment. You mean?
Yeah. Yeah. No, normally the yeast would grow better at what is the optimal temperature for the yeast to grow? That 30, 37.
It is said that yeast can grow up to 42 degrees.
Okay, and you can define as the thermotolerant.
Yes.
Okay, all right. Why are you interested to isolate the thermotolerant yeast from flowers? Why flowers?
Because previous research, according to previous research,
use the forester to coming there and plant the seeds in the wildfire impact areas. So that is we use high cost and we use many person to go there. But we have the drone. The drone can flying across the forest or anything. Yeah, we can easy to going their area. That is can helping the forester. Yeah. Yeah, it's use this innovation is more than a human. Yeah.
Okay, so in one of your experiment that you have non-treated seed and you have the hydrogel, what is the non-treated seed and how did you compare them? Yes.
I'm just using the seed. I want to compare. It's like a normal planting. We compare using hydrogel seed pod and normal planting.
So you have to go to that area, the same area.
Yes, it's the same. We testing in the same area.
Yeah, I just wonder how did you control the area of the experiment? Because you put the drone, it's very high, and you have to sort of release them into the soil, right?
Yeah. I control by using the same altitude and the same places to test the experiment.
So then after you plant it by the drone, you have to go to that area and look at the seed. Whether they germinate or something.
After we sorry.
Because our law that we can fly the drone in some place that after the wildfire place, so we just test this in our home areas.
Oh, in the home area, not the wildfire
because
area.
of our law that we can fly the drones and it's difficult to ask them to can we test this.
So, but you use the drone to do the experiment, but in your own area, just like a small area. Oh, okay. I just wonder how can you do it for real? Okay. So could you go back to the hydrogel seed pot? Yeah, do you have the picture of that? And yes, yes, this one. So could you explain a little bit in terms of the innovation that you have done? Yes. You have outer and inner. Yes. What are those and what? Yeah. Why it has to be this way? Yes.
Let me explain about this.
Yeah.
Okay.
Okay.
So in the from this factor, the soil is the most important for the for helping the seeds can going in the wildfires. So you can see this picture. The soil lost the humidity, lost nutrients for helping the seeds can going in the wildfires impact area.
Okay.
So let's see the features of of this hydrogel seed pod. We using the PVA solution and calcium sulfate is the outer of the hydrogel seed pod, and the main of this hydrogel seed pod. We using hydrogel polymer and nutrient that helping the seeds can going in the wildfires. The feature of the hydrogel is can absorb the water. Yeah, that is helping the seed can going in the wildfire that lost some humidity. Yeah.
So is it the closed system? I mean, if you have this and you put into the soil, and what happened after that? How did the seed would be released from the seed pot?
Yeah. The seeds will be released from the pot when it touch the water.
It touched the water.
Yeah. It will.
So it would stay like that.
Yeah. This when this when the water touch the hydrogel seed pod. by
okay.
using PVA solution that is can water soluble. So that is make the pot is spread out. Yeah.
Oh, okay. So the seed would be waiting until there's the water come into that soil area.
Yes.
Oh, okay. All right. That's interesting. That's interesting idea.
Yes.
So did it work?
Yeah, this is work. Yeah, this work.
It worked. Okay.
Okay.
But you have to select like the type of the seed, right? It
Yeah, yeah.
depends on which area that you want to
Yeah.
put this seed in.
Yes, we study if you want to reforestation, you want to know the type of seed that the forest area that we
Yeah.
want to repair the forest.
Yeah, and how stable are the seed in the pot? How stable?
How stable?
How long did it last? 1 year? If there is no water at all, you do still release and germinate. Yeah, it's just a question.
Yeah, I think about a year.
A year, okay. But I think it depends on the type of the seed and how well of the gel would work to protect the seed.
Yes.
Okay. All right, thank you very much.
Yeah, thank you for attention.
Thank you for your feedback, commentator. We would like to invite our commentator, Associate Professor Dr. Vilaylak Sriphon Dulasin from onto the stage to give the certificate of presentation. And the last presenter are from Tennoji High School attack to Osaka Kyoiku University, which the project title state to the land in 30 grades forgot on their protein. Please come to the state.
Hi, my name is Amane Fujiwara.
And Yuna Kawai. Our research theme is stress tolerance in tardigrades focused on their proteins. Tardigrades, also known as water bears, has 8 legs and length about 1 millimeter. It is known for its ability to alive in dry conditions. In this state, they cannot die. They do not die even if there are various stressful environments such as high or low temperature, vacuum or high pressure, cosmic radiation, and X-ray. Previous research has shown that Cas protein, which is expressed only in tardigrades, is related to stress tolerance. However, the relationship between tolerance and proteins that many organisms have is not clear. Therefore, we focus on GAPTH protein. GAPTH protein has a function of glycolysis, which is also preceded by eukaryotes such as humans.
The reason we focus on this protein is that tardigrades have this protein more than other proteins. Therefore, the purpose of this protein in tardigrades is more thermotolerant than GAPTH proteins in other organisms. To analyze GAPTH protein, it is necessary to purify and get only GAPTH protein in tardigrades. However, tardigrades are too small to acquire the protein directly from individual tardigrades. So we use E. coli. E. coli has function of doubling in number in 20 minutes. Putting the GAPTH into E. coli, we can get a lot of GAPTH. Therefore, we first amplify the insert and vector. We proceeded with the experiment in this order: cloning GAPTH gene from tardigrades, protein purification, and enzyme activity. We first removed 10 tardigrades from the agar medium using a micropipette and ground in individual tardigrades. From there, we extract mRNA using a kit, obtain cDNA from the mRNA using RT-PCR method. Next, PCR performed to amplify the insert GAPTH gene and vector PET28A. PET28A is a type of plasmid. Here is a PCR condition and primer array. We checked that PCR was successful by electrophoresis. When GAPTH and PET28A have the same sequence, the two can be linked using enzyme recombinase. Then, here is the vector map of PET28A. Then, E. coli was transformed by heat shock method, in which the vector was incorporated into E. coli by a rapid temperature change, and the plasmid with the GAPTH gene was incorporated by the infusion reaction. The plasmid was used because the GAPTH gene alone is not taken up by E. coli. After 1 night of incubation, colonies are expressed.
This is the picture of colonies, and to make sure these are recognized, we did CAR-check by using agar-scale electrophoresis. And blue arrows point recombinase recombinase plasmid, and the yellow arrows point the PET28A, which is the plasmid only. The longer plasmid goes slowly and stop soon, which is when it compares to the short one. So this is successful to recombinase. And we purified the protein. First, we broke down the cell walls by using ultrasound. And how to purify the protein is using the column to correct GAPTH protein. And we measured the enzyme activity using the GAPTH protein. And GAPTH protein has a GAPTH protein make GCP to bisphosphoglycerate. Then when it turns bisphosphoglycerate, the NAD become NADH the NAD is colorless and NADH is yellow. So it turns yellow, the GAPDH works. And how we measure the enzyme activity is here. And then we export to light at a wavelength of 340 nanometer. And we change the amount of G3P. And this is the result. And this is the conclusion of our research. First, we created E. coli and expressing GAPDH protein and purified the GAPDH protein. Second, the enzyme activity of GAPDH protein was measured. So what we want to do next is measuring the enzyme activity of the tardigrade GAPDH protein at different temperatures. These are the acknowledgement and references. Thank you for listening. Yeah. Oh yes. First, we tried to, we tried to do like different temperatures, and next, compared to the other animals, like such as
It is because room temperature. Yeah.
like how we get a like what do you mean, how we get a GAPDH, like RNA that we grinded the tardigrades and then use the kit for the like we can get the match RNA from tardigrades, so we did we used E. coli to make you can see the that tardigrades only in Hokkaido in Japan, and we couldn't go Hokkaido, so the teacher gave us the university teacher gave us the
Yes, that's a nice presentation. So is the tardigrades is the animals, right?
Yeah.
Invertebrate animals. Is that right? Invertebrate. No spine, no spinal.
Oh yeah.
Yeah, so it'd be invertebrate. So in your experiment, you did the GAPDH from tardigrades, but in your abstract, you mentioned about the human too. Yeah, so any correlation between this protein of tardigrades and humans?
We wanna research about that, and like the human GAPDH was researched already, so we can know the structure of the gap, like no, the humans GAPDH, but we never know the tardigrade GAPDH structure.
Okay,
Yeah.
all right. So in theory, this protein of tardigrade should be more effective than human, right? Because we cannot like survive through the very harsh stress, right?
Maybe.
Yeah. Yes, maybe. And so I just wonder why this protein could be working well under the dry condition. What kind of function of this protein?
GAPDH protein.
Yeah. Why this protein is important for the dry condition of this animal? Yeah.
As a GAPDH protein has a function of glycolysis.
Glycolysis. So it's normal, right?
Yes.
It's normal, yeah. So
Yes,
why?
it is also has a function of to become and yeah.
To make cells die, or like
Oh, okay. So make them to make the cells survive under dry condition.
die.
Die.
Yeah.
Or die. Dead. Or dead or die.
Yeah.
Oh, okay. So any information the difference between this protein between tardigrades and humans?
We will compare the structures.
Yes.
Like we made the crystal of GAPDH.
Okay. Yeah, you do have information. What are the difference?
We didn't research yet, but the amino acids, like
Amino
the-
acid.
yeah, amino
Oh,
acids.
okay. That could make them function differently. Ah, okay. So you did the cloning of this gene in the E. coli, and you sort of extract the protein from the E. coli, right? But in terms of the function, because it's the eukaryotic genes, right? In E. coli, does it work properly?
Oh yeah, like E. coli is easy to increase the number, so we use that. Like, yeah, it is difficult.
Oh, okay. Still on the way, under investigation. Yeah, okay. All right, thank you very much. Sorry, its timing is over.
Okay, thank you for the feedback, commentators. We would like to invite our commentators, Dr. Hatapak Deshakaisaya, onto the stage to give the certificates of presentation. Everyone, please come on to the stage for take group photo.
with us in Smart Card Room 2304, scheduled for the presentation under the theme of Biology 3.
We would like to welcome you all to this 16th project oral presentation. First of all, we would like to introduce our commentator. The first commentator is Associate Professor Dr. Wilaylakshmi Siriporn Dulasin from Khon Kaen University. And we also have Dr. Hattapak Desha Kaisaya from Princess Srisawangkawat College of Medicine, Julapon Royal Academy as our second commentator.
We are also happy to introduce 6 schools that will be joining this oral presentation. The first, Princess Julapon Science High School, Buriram. Check ชื่อค่ะ Next, Tokyo Gakugei University Senior High School. Next, Princess Julapon Science High School, Pathum Thani. Next, Shizuoka Hita Junior and Senior High School. Next, Princess Julapon Science High School, Chiang Rai. And the last one, National Institute of Technology Kosen Tsurima College. To begin the sessions, we would first like to explain the guideline of each presentation. The time for each presentation is 15 minutes, 1 minute for preparation, 7 minutes for the presentation, 5 minutes for comments and discussion, 2 minutes for certificate and take photo.
Please check the time shown on the monitor over there. If I show this sign, it means there is 1 minute left. If you see this sign, it means that you should stop because time's up. At the end of the oral presentation, every presenter will receive a certificate of presentation, and we encourage everyone to come together in your respective for your group photo sections. During the presentation, please with your mobile phone. If you want to take photos, then please stay in your seat and refrain from walking around be mindful of the audience.
Now, we will start with the first group from Princess Julapon Science High School, Buriram, and their project title is Development of Fruit Coating Foam Colorway for Inhibiting the Growth of Cholecdotrichum, SPP, and antioxidant properties in golden Namai mango fruits. Please come on to the stage.
Test. Greeting to all guests of honor, professors, teachers, and friends. We are the representative from Princess Chulakon Sai High School, Buriram. My name is Sitkanya Panyadi.
And my name is Penpicha Thasima.
We are here to present our science project, development of fruit coating from caraway for inhibiting the growth of Coledoxylum species and antioxidant property in golden Namdokmai mango fruit.
This graph shows that our country, Thailand, earns a lot of income from exporting mango, and it tends to expand more and more in the future. The most exported species is the golden Namdokmai mango. Thailand also export mangoes to Malaysia, South Korea, Japan, and China, but the mangoes can be damaged from the long transportation, and it can be noticed by the black area on the mango skin. We'll show its symptoms when the mango has begun to ripen, and will spread as the mango gets more ripened. This disease called anthracnose, a fungus that can cause the disease is called Coledoxylum species. This disease destroys more than 470 species of legumes, vegetables, fruits, and ornamental plants, which had a huge negative impact on developing countries like us. In this present time, the use of chemicals in the group azoxystrobin, borax, and benzimidazole is to resist diseases, but the chemicals in this group are dangerous to the ones who really touch and eat them. According to Xuan Zhesheng's research, carbon that's extracted from the essential oil of peppermint is effective against this fungus and has antioxidant that helps slow down the ripening of agricultural products. From the study, we found that caraway, a Thai herb, contains more than 44.5 to 95.9% of constituents in essential oil called carbon. Shellac fruitcake is a type of edible coating that has been approved by FDA. Moreover, our country is an important source of shellac in the world. Thus, we are interested in using the essential oil that extracted from caraway seeds to evolve it into food skin coating matter to inhibit the growth of Coledoxylum species and slow down the ripening in golden Namdokmai mango, which align with sustainable development goal number 3, good health and well-being, and goal number 12, responsible consumptions and productions. The hypothesis of this project is the food coating from caraway can inhibit the growth of Coledoxylum species and slow down the ripening of the golden Namdokmai mango. In terms of the procedure, we divide into 2 parts. In the first part, we prepare the food coating. In the second part, we test its properties. Start by extracting the essential oil using hydrodistillation. After that, we used FTIR method to detect carbon, and we found it to contain carbon.
Okay, then we will test the caraway essential oil's antifungal property by using the method called poison food technique. We will mix the taste substance, in this case caraway essential oil, with the agar media and pour it into a petri dish and let it solidify. Then we will press and plug up a piece of the agar that contains the fungus, put it in the plate which we have prepared earlier, and put them in the incubator at the room temperature for 2 to 3 days. Then we will use the vernier caliper to measure the colony diameter of the fungus and calculate the percentage division using this following equation. Okay, let's go forward. And the result showed that the caraway essential oil possesses an emission percentage of 73.08. Then we will start preparing our fruit coating. Our fruit coating will contain 3 different solutions. The first is caraway essential oil solution, can be prepared by dissolving caraway essential oil in sucrose ether solution, which is an emulsifier. And the zeolite solution, which can be simply prepared by dissolved zeolite powder in water. And the last is shellac solution, which can be prepared by dissolving shellac flakes in diluted ammonia and dilute it once again with water. This is a prototype of our product. And you can see that there are 3 different solutions, so we must find which one is the best solution. We will divide our fruit coating into 7 different ratios, including
the ratio of colorway essential oil solution to silane solution to shellac solution in 1 to 1 to 1, 1 to 1 to 2, and it goes on. Here is our result. Okay, then we use the same method to find the fruit coating's antifungal property. And the results show that the fruit coating number 2 has the high percentage emission of 22.55. Then we want to find out about the fruit coating's efficiency in antioxidant property. So this will be divided into 2 parts. The first is color measuring, and the second is weight loss measuring. Okay, first of all, we will draw a 5 circle over the mango skin to indicate an area in which we will measure the color of the mangoes. Then we will coat our mango with our fruit coating. Okay. Now we use this device which is called colorimeter to measure the colors, the color value of the mango skin. And we use the data we collect from the colorimeter to calculate color different number. This number will tell us how the color of the mango skin have been changed from day to day. Down here. And we can see that the fruit coating number 2 has a different number of 15.64, which, there is an graph version. So if you see the fruit coating number 2 is the yellow line, and there will be the control, which is the mango that hasn't been coated with anything. So we can see that fruit coating number 2 has the better result than the control mango. The last part is weight loss percentage. So we also weigh the mango 3 times each to calculate weight data and calculate weight loss percentage. Using this equation, we will see how the weight of the mango has been lost throughout the 7 days experiment. And the results show that the fruit coating number 2 has the low weight loss percentage of 10.12. Here is also the graph version. Result and discussion. So in conclusion, from our study, we have found that the fruit coating contains colorway essential oil, which contains carbon substance, can be cholesterol species, and the best ratio was found to be the ratio number 2, with the high percentage emission and the lowest weight loss percentage. So even though it didn't show the lowest color different number, but the ripeness of the fruit isn't solely determined by the change of the color, but it depends more on the weight loss percentage, which relates to the softness of the fruit. Since when the fruit began to ripe, it metabolism and expel out the water, thus making it lose its weight. So if our fruit coating has low weight loss percentage, it means that it is a good antioxidant agent against the fruit, since it will help reduce the metabolism rate inside the fruit. Yes, other thing. Thank you for your attention. Feel free to ask any question.
Thank you for your excellent presentation. Next, I invite our commentator to give advice or comment about this project.
Okay, thanks for your nice presentation. Very colorful of the mangoes. So what is the normal approach that they use for the coating, the fruit coating, like mangoes? Did they use chemicals or something? And do you have that as the control to compare with your colorways coating?
Well, as we said, there will be a chemical in group like azoxystrobin or brocolate, but there is a one we use as a control. It's called carbendazole. Which one? Maybe right here the last.
The last one.
Yeah.
The carbendazole plus PDA percentage inhibition is 100%.
Yeah.
So chemical is the best in terms of inhibition.
Yeah, but it can be dangerous if you not use
in terms of safety. Okay. And when you test the antifungal activities of the colorway coating and the fungal, the purified strain, did they show the same result? I mean, the antifungal activity has been reduced or not after you prepare them as the colorway coating? Because you have the first experiment that you test the carvone, what's carvone compounds in the oil with the fungal. Could you go to the slide that shows the result of inhibition? Yes, I kind of remember when you did the test that you measure the colony diameter.
This one.
This one, this one.
This one.
Yeah, okay, so the best inhibition is about 73%, right?
Yes.
When you have, I would say, maybe 1-to-1 ratio. So what about the colorway coating after you prepare? Did that show the same result, or the inhibition activity was reduced?
Yeah,
or
it
not?
was reduced.
Ah, okay.
The reason behind this, maybe there are the solution isn't mixed well, and there may be anti-against each other in some parts. We don't know that actually, and we would like to find out more.
Okay, because we don't know whether the combination would be optimized or the best for the activities. And my second question is about the antioxidant activity, that you use the color as the indicator. So how the color, different color, would tell us that there is the antioxidant activities?
Well, in a normal sense, when the fruit ripens, the skin obviously change. The unripe is green one, the ripe is yellow one. So it's basic as that. And for the scientific explanation, so the fruit is skin color can change from the oxidation reaction inside the fruit. It's one of the indicator. But yeah, well, maybe the color isn't the best indication of the ripeness of the fruit, because there are many factors that can affect the color of the fruit. We imagine if there is a wound, open wound in the skin, so the color can change despite their ripeness.
Yeah, and also you mentioned about the weight loss of the skin. If the weight loss is reduced, that would mean it's not that good. Is that right? When the weight loss is reduced, it's not good.
Oh no! If the weight loss is reduced, it means it's good. Yeah. So the mango will have the same weight like before. Okay. I explain it. Sorry. So when the food arrives, it metabolism, and throughout the process they expel out the water, making it lose its weight. So to slow down, it might have the low weight loss percentage. Do you know what
All right, very good presentation and very detailed explanation of the methods. For the color, for testing the color, did you use the chemical coating to test as well?
Oh no, sadly.
And also, when you were picking the mango, did you pick it from the same batch, or how do you measure the baseline for the mangoes?
Yeah, we order the mango from the same farm. We tell the farmer that we want the mango to be the same as possible, like in size, in colors, and maybe even from the same tree. And we ask them to not coat it with anything when they are send it to us.
Very good. And will this be applicable to other fruits as well?
Yeah, we hope so, but we actually we didn't have time to experiment on the other fruit, but we hope we can develop to use in the other fruit as well.
Thank you. Did you have to eat a lot of mangoes?
You can try another types of mangoes, not just Suriname dok mai. Yeah. Okay.
Thank you for the feedback, commentators. We would like to invite our commentators, Associate Professor Dr. Vilailak Siriphorn Durasin, onto the stage to give the certificate of presentation.
Thank you, the commentators and presenters. The next presenters are from Tokyo Gakugei University Senior High School, and their project title is Relation of between sunflowers, heliotropic movement, and auxin. Please come on to the stage.
Thank you. Hello, I'm Mariama from Tokyo Gakkai University Senior High School. I'll talk about my research relation of between sunflowers' heliotropic movement and auxin. Introduction: young sunflowers track the sun. It's called heliotropic movement. You know, most plants shows phototropism like this. The movements are also caused by auxin. My hypothesis is that auxin probably moves differently between sunflower stems and the stem of other plants. I conducted 2 experiments. First, I did an Aveno test to determine the amount of auxin in different parts of the sunflower stem. Second, I took time-lapse photographs of heliotropic movement. First, Aveno test. Aveno test is micro-determination methods for auxin. First, I prepared a sunflower and marked and cut the sunflower in this way. Also, I cut the stems to the same size. Next, I made 1% agar pieces to fit the stem pieces, and I placed the stem pieces on the agar pieces, and then stored that in the dark for about 24 hours. Also, I prepared a spirit of pet plant like that, and I attached the agar pieces on them, such as that, and stored the shoots in the dark about 5 days. Finally, examined the degree of curvature of the pet plants. This table shows the result. I analyzed based on previous studies. First, the concentrations of number 1 and number 2 are higher than number 3 because number 1 and 2 were bended. Second, the concentration of number 1, which is bent in the direction of the agar piece, is higher than number 2, which is bent in the opposite direction of the agar piece. So we can see that the auxin concentration is high in the order of 1, 2, and 3. Therefore, auxin concentration in sunflower stems is highest at the top of the stem and decreases toward the bottom of the stem. In other words, there is a correlation between auxin concentration and heliotropic movement. Moving on to experiment 2, I fixed the camera like that and took photos of the tip of the sunflower in a time lapse at 10 minutes intervals. There was a greenhouse, so they were not affected by wind. This is the information about the location and the date and time of the shooting. Yeah, this is a movie that connects some of the pictures taken. This is the result for the angle of rotation. I classified them in this way. This table shows that the greater the angle of rotation, the longer it takes to rotate. Also, the smallest number of times was the small rotation. Next, I found there was no look at the table. I found there was no significant difference between the clockwise one and the counterclockwise one. It means that movement doesn't track the sun. This is the table of the whole day rotation of the sunflower. On most of the days, there are more rotation at night. From these results, I think the movement is not heliotropic movement. Also, I can think sunflower have not only heliotropic movement, but also this movement. This is conclusion of my research. There is correlation between heliotropic movement and oxygen concentration. Sunflower has not only heliotropic movement, but also other movement. If oxygen is the cause of this movement, oxygen is rotating through the sunflower stem at a fast rate. Here are acknowledges. Thank you for listening.
Thank you for your excellent presentation. Next, I invite our commentator to give advice or comment about this project.
I find this movement. I think sunflower has other movement. So I want to find another movement and more. Yeah.
And this is a new finding, right? Like no one has identified this rotating before, right?
Yeah. I couldn't find on the previous studies. Yeah.
Why do you think it moves like that? I think I saw at night it moves faster or something,
Yeah.
right? Yeah. Why do you think it do that?
Sunflower, a heliotropic movement to sunflower do heliotropic movement to more grow up. So this movement too, or some or yeah, I don't know. Sorry. Yeah.
Yeah, I think that's sort of interesting. I never heard about this kind of study. So the heliotropic is the move to the sun, right?
Yes.
What about the light? Yeah, but so when you grow them in the greenhouse, do you have the light around area and when at night? Maybe that's why the plant would move faster. I'm not sure. Just
Okay. Yeah. There is just sunlight. So at night, not there isn't something like.
just very dark.
Yeah.
Oh, so yeah, I just wonder why they move faster, or they try to find the light, to move to the light. Yeah.
It's
Yeah.
just dark.
Yeah. So have you measured anything? I mean, like oxygen at night or at day, and you see the difference between night and day.
Oxen. Oxen cause the phototropism. So oxen react to the sunlight. So I don't know, but heliotropic movement is sunflowers time. It's human time, like a human's time. I don't know. Yeah. So yes. So oxen. I don't know, but oxen react sun and sunflowers custom sunflowers life work rhythm. So yeah.
So heliotropic has nothing to do with the oxygen. It's not related to the oxygen concentration. Is that right?
I don't know. Yeah, I think heliotropic movement and there is correlation heliotropic movement and oxygen concentration, but yeah, I say but oxygen the tip of the stem make the oxygen a lot of, so the result is the result is maybe correct, I think, but yeah.
So you have to measure the concentration of oxygen.
Yes.
That might be help. And my last question: why sunflower? Why are you using a sunflower as the?
Because I like sunflower.
You like sunflower?
Yeah.
Okay.
Yeah, I like sunflower since when I junior high school. So yes.
All right.
Sorry.
So it's very beautiful flowers. Okay. Thank you very much. Okay.
Thank you for the feedback, commentator. We would like to invite our commentator, Dr. Hattapak Deshakaisaya, onto the stage to give the certificate of presentation.
The next presenter are from Princess Julaporn Science High School, Pathum Thani, and their project title is Development of Prostate Cancer Antigen PCA3 Test Kit in Urine by Nucleic Acid Hybridization Technique predict the risk of prostate cancer.
Hello everyone. We are students from Princess Julaporn Science High School, Pathum Thani. My name is Arinthorn Bunmat.
And my name is Thanabudi Kasian.
Today we will present our project about development of prostate cancer antigen 3 test kit in urine by nucleic acid hybridization technique to predict the risk of prostate cancer. Prostate cancer is the second most found cancer in men and is the fifth most common reason of death in the worldwide population. It is commonly tested by measuring the amount of PSA in blood. But rising levels of PSA does not only occur in prostate cancer but also in other conditions like BPH, which results in low specificity for prostate cancer. And from the low specificity of PSA, there have been studies conducted to find better biomarkers that have more specificity, which is found that PCA3 in urine is also another important biomarker. And moreover, there have been studies about using gold nanoparticles as biosensors by using nucleic acid hybridization technique. And this method has many advantages, such as safety and convenience. And by using this technique, an easy-to-use test kit can be developed, thus leading to our project to develop a prostate cancer antigen 3 urine test kit by using nucleic acid hybridization technique to predict the risk of prostate cancer.
The variable blinding test of PCA3 with the label probe, independent variable amount of PCA3, dependent variable color of solution, control variable amount and concentration of buffer, amount of probe of conjugation, testing temperature, and testing duration.
Firstly, we analyzed the nucleotide sequence of PCA3 to find the suitable probes. We downloaded the nucleotide sequence from NCBI, and then we used the PrimerBlast tool to find the nucleotide sequences of the probe. Then we download the sequences and then sent it to YouTube Bio to synthesize. And next, we cultured our prostate cancer cell by using PC3 cells and F12 medium. After that, we extract the RNA from our PC3 cells, and then we converted the RNA to cDNA. After that, we use the cDNA that we have as a DNA template in RT-PCR by using the primers in the reaction as shown in the table. After that, we conjugated the probes that we synthesized with gold nanoparticles. And then we did a binding test of PCA3 with the labeled probe.
The results: first, analysis of PCA3 nucleotide sequence to find suitable probe. The probe obtained from the PrimerBlast is shown in the graph. Second, preparation of prostate cancer cell culture. From culturing PC3 cell in culture dish, cell is increased in number and adhere the cell culture surface. And the last one, binding test of PCA3 with the label probe. From combining conjugation, solution of the sample, it's found that color solution with PCA3 has change in color because the gold nanoparticles are larger. And when we analyzing color image of conjugating solution by histogram in image, the value of conjugating solution containing PCA3 value is shown in the graph. The conclusion: nucleic acid hybridization can be used to detect PCA3 in the sample. When we detect the PCA3, the solution has change in color. This method can be used to develop PCA3 test kit in urine to predict the prostate of cancer in the future. Thank you.
Thank you for your excellent presentation. Next, I invite our commentator to give advice or comment about this project.
PCA free. Usually, when they suspect that they might have prostate cancer, they usually already have cancer when they go check because they've been having problems with the urinary system. So what our test kit aims to do is to have an early detection, which is non-invasive. We don't need to draw blood. We just urinate out and then measure and look at the color.
Okay, and I imagine the level of this biomarker in the urine will be a lot lower than in the blood, right? Will it still be detectable? Especially if you're going earlier than when you're measuring the blood as well.
This project is our proof of concept in this method. Is our result. It has.
So it can detect, but you don't know what's going to happen in the urine yet. Okay. Okay.
Yes.
So how did the color could be developed? What did you put in this tube? In this tube, what's in there? Yes.
So the colors are from gold nanoparticles. So this one is about 20 nanometer gold particles.
It's a gold particle. Okay.
Gold nanoparticles. And then when they conjugate with the probe, and then the probe connects with the PCA3, it becomes a bigger piece. And then gold nanoparticles, because they are very small, they absorb only some part of light waves. And when they get larger, the color changes because the absorbance unit changes.
Okay. And how did you prepare the sample from the urine?
We didn't really get urine. We synthesized our own solution that has PCA3 by mixing it with a buffer.
So haven't used the urine yet?
Yes, not yet.
Okay. So in terms of practical terms, so if you have the urine from the patient, what you have to do this test? Do you have to correct the cells? Do you have to extract the DNA? Or yes? Yeah. Just in terms of practical. If you are going to use it, how would you do it?
If we develop it.
Uh-huh.
Yes.
Okay. Oh. Okay,
We,
okay.
we
So
have
it's the
sample
urine,
is
you have the
urine,
buffer,
yes,
buffer,
and put
gold
it
particle.
together, and
Okay. How do you make sure that the urine already have the DNA in that? Because it's the cell, right? So that the cancer cell just would be released into the from the blood to the urine, and you don't have to do anything with the urine at all. How the nucleic acid would be come out from that cells? That's my question. Yes. Yeah, if you have just the urine and you put the DNA probe, whatever, the gold nanoparticles, how would the reaction would occur?
Berseria that change when exposed to different wavelengths of light. Please come on to the stage.
Test.
Thank you. Sorry. So the response of the Paramecian versus the changing when exposed to different wavelength of light. Paramecian Bruceria protease take chlorella into their cell and live in intercellular symbiosis. It can also survive and re-symbiosis once it has lost symbiosis chlorella. The Paramecian Bruceria provides carbon dioxide to nitrogen to chlorella for chlorella provide oxygen and sugar to Paramecian Bruceria. In a nitrally vicary symbiosis relationship between the Paramecian Bruceria and chlorella, we were intrigued by the behavior of Paramecian Bruceria in response to light and began to study them. The experiment was based on the result in three studies considered of the last year. The Paramecian Bruceria and Paramecian Campdots were provided by Yamaguchi University. We have been studying absorbed spectrum of Paramecian Bruceria when exposed to different wavelengths of light. And the Paramecian Bruceria, and the response of Paramecian Bruceria when exposed to different wavelengths of light. We began our search with the two hypotheses. One, chlorella absorb blue, violet, and red light, and chlorella exhibition positive hot axis to blue, violet, and red light. First, we measured the absorption spectrum of the Paramecian Bruceria. A Paramecian Bruceria when placed in the cell, and the absorption spectrum were measured using spectrum photometer. This is a result of the violet and blue light absorbed, but red light was not. Next, we measure the absorption spectrum of the bleached Paramecian Bruceria. This is a result of the violet and blue light was absorbed, but red light was not. Next, we measure the absorption spectrum of photosynthetic pigment of chlorella. This is a result. We found the red light was absorbed less than violet and blue light. Next, we measure the absorption spectrum of the photosynthetic pigment in single chlorella. The experiment was periodic using column chromatography, which although preparative separation in the stage of the component or substatics. Chlorella and tablet from which is commercially available was used. This is a result. This is a result. It was found the red light was as well as blue and violet light were absorbed by the photosynthetic pigment in single chlorella. Single chlorella absorb red light. We also consider the chlorella use red light or photosynthetic. The research so far was led us to wonder if red light is not absorbed by chlorella in the body of the Paramecium brucellea. We therefore consider the photosynthetic experiment. Petri dish with 100 Paramecium brucellea cell, a 900 microliter culture medium. One LED bulb was placed on the right side of Panacua stereoscope. A Petri dish was placed a distance of 5 cm of LED bulb, and the number of Paramecium brucellea cells going the opposite direction of the light was counted. This is a result. Negative traveling prepares were observed in the red light. The Paramecium brucellea, the hot axis of British Paramecium brucellea was examined. The experiment was similarly on the exchanging British Paramecium brucellea two different wavelengths of light and measuring the number of cells in the British Paramecium brucellea as they moving away from light. This is a result. Negative traveling prepared was observed in the red light. Next, we also examined the hot axis Paramecium camudazme. This is a result with the Paramecium brucellea negative learning was observed in the red light. From the result of the experiment, we wonder if the Paramecium camudazme and Paramecium brucellea had negative learning to feel from red light. Therefore, in individuals with the behavior of Paramecium camudazme changing way exposed to the two type of light. In the experiment, we will draw a line developed as a case to 3 equal parts. Individual to red and blue are the same instead from the both ends, and measure the number of cells in the individual that the favor toward blue, that light, those toward red light, and those that were discount toward neither. This is a result. Both red light and blue light were found to have effect with the more individuals moving toward blue light and fell toward red light. A certain number of A certain number of individuals was also found the midpoint where both red light and blue light. And from study after 2020, the number of cells increased in white light, violet light, and blue light, and did not increase much in yellow and red light cell. Cell counters increased in white and violet and blue light, and did not increase much in yellow and red light. And the amount of pigment in chlorella did not change when exposed on wavelight. The growth rate of the red light was similarly on the blue and purple light, and the number of chlorella cells was almost considered within the Paramecium brucellea. Existing studies have also showed on the photosynthetic pigment in burnt, absorbed blue, violet, and red light colors, and use them the photosynthesis. And single chlorella absorb red, blue, and the red light. Bleached Paramecium brucellea absorb violet, blue, and red light. And we found the Paramecium brucellea and bleached Paramecium brucellea are positive hot axis on the blue light. Both Paramecium camudasme and Paramecium brucellea were negative hot axis to red light. This led Paramecium brucellea to believe the chlorella may be the splitting photosynthetic with red. We then considered the positivity that Paramecium brucellea will provide the increased chlorella from absorbing red light, because the amount of red light absorbed by the increased chlorella are low, and photosynthesis was not taking place. We also spectrumed the negative hot axis to red light was due to the diurnal behavior of fish, which freeze from surface of the water in the evening. Therefore, we saw the might altering intracellular chlorella escape to light. That's conclusion my presentation. Thank you for listening.
Do I have time for questions? 5 minutes. Oh, okay. Yeah. Okay, 5 minutes, right? Okay. So I summarize that both the chlorella and Paramecium prefer the blue color, right? The blue light, the blue wavelength. Blue, right? Yes, blue light. Okay, all right. So why is the blue light? Yeah, so why both Paramecium and chlorella prefer to respond to the blue light? But they have different pattern. Okay. Yeah. Yes, the blue light. Yes, blue. Yes. Why? Why is blue? Uh-huh. Yes.
uh, 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 Plant absorb blue and violet and red light photosynthesis.
What about the paramecium? So in your experiment, do you have like different cells? I mean, you have just cholera, you have just paramecium, or you have both of them together? That I think the response could be different. So if you have just cholera, the clean colors, yes, the algae, and you have just paramecium without cholera, or you have both of them, will they respond differently to the light, to the blue light? Okay, maybe it's okay. All right, it's okay. Just yeah. If you look at the pattern of the light response of the paramecium and the cholera, I see the different patterns. So you have the graph shift at the blue light and then go down and it's stable and shift again. But for the paramecium, you have the light at the blue light is very high and then drop, decrease continuously. Yeah. So I just wonder why it have this type of patterns. Yeah, light response patterns, they look different. Okay, is that okay?
We also spectrum the negative phototaxis to red light. We was the diurnal behavior of fish, which freeze from surface of the water in the evening. Okay.
Thank you for the feedback, commentators. We would like to invite our commentators, Dr. Hatapak Deshakaisaya, onto the stage to give the certificates of presentation. The next presenters are from Princess Juraporn Science High School, Chiang Rai, and their project title is Cellulose Enrichment from Fermentations with Various Carbon Sources. Please come on to the stage.
Good afternoon, everyone. My name is Kanokwan Sae Huang.
And my name is Jalavi Chantharaporn.
Today, we would like to present our project, named called Cellulose Enrichment from Fermentation with Various Carbon Sources. For introduction, cellulose is commonly found in plants and bacteria, but bacteria cellulose will have better properties than plants due to the unique properties of good bacteria cellulose, which are often used in medicine, such as the treatment of certain burns and treatment of burns, absorbing oil on the sea, and making composite material to trap heavy metals in the water. Cellulose bacteria can increase by tissue culture and the fermentation, but the tissue culture of bacteria cellulose is costly and well equipment. On the other hand, kombucha fermentation contains clear agar that is not often used and left over, in which there will be bacteria cellulose. In this way, the cost is lower than tissue culture, and then can use waste material to make use of it too. And our objective is to study the suitable carbon sources for increasing cellulose by kombucha fermentation. And the second is to test the properties of cellulose derived from kombucha fermentation.
For PEG experiment, we have two steps. Step 1 is material preparation and fermentation of various carbon resources. First, we have this flora, carbon source, water, and SCOBY. We have 7 carbon sources. It's pineapple, sugar banana, and sugar cane. We will wash and cut it like this, dice, and then boil in the water, let it cool, and mix. Follow formula, and the set for carbon source is pineapple peel, pineapple leaf, sugar, and the last is sugar and tea. We will crush it to be the powder and then boil in the water, mix with the hot water, and then follow from formula. Next, we will measure the pH level. If it more than 4.5, we will adding the vinegar until it less than 4.5, and then cause it with cheese coat and loop. Plant it in an area out of direct light for 12 days. After 12 days, we will got the cellulose. We will next the step is a cellulose property test. First is dry wet test. We will wash it 5 to 6 times and then boiling in sodium hydroxide for 4 hours. Then soak in hydrochloric 13 minutes. Wash in water until pH level was neutral, and then dry until we can weigh the cellulose. And the next is water retention value. We will weigh the dry cellulose and then soak in room temperature for 3, 6, 9, and 24 hours. Rot the surface area and then weigh the cellulose in 3, 6, 9, and 24 hours. Next, we will be beta cellulose content. We will soak the cellulose in sodium hydroxide for 13 minutes. Then mix with water and wait for 13 minutes. Filter out of ceramic and then prepare resort solution and put in the stopper test tube. Mix with the sulfuric acid and dip in hot water for 2 and 3 minutes. Then centrifuge until we got the ceramic. And the last is structure cellulose content. We will find the structure in the microscope under 100x magnification and record.
For result and discussion, the water retention Viral tests show that the cellulose from kombucha fermentation using pineapple peel carbon sources had the highest percentage of water retention value. This is consistent with the results of Thanyapon 2014, where the materials absorb water well when it has a hydrophobic function group and tends to allow molecules to diffuse. And pineapple peels have the best properties than other carbon sources.
Using banana as a carbon source, your high density fiber based on low cellulose water retention value. This is consistent with the cellulose structure under a scanning microscope of Jutharat Maukapneut, who produced cellulose bacteria with Actobacter salinum by using pineapple processing with water as carbon source and expanded when the structure of the cellulose has a high density. It will result in less water retention value. Determine cellulose result indicate that cellulose content in SCOBY from kombucha fermented by carbon source, pineapple peel and banana. Thank you very much.
Thank you for your excellent presentation. Next, I invite commentator to give advice or comment about this project.
Okay, thank you for the presentation. Do you mind explaining the rationale behind the hypothesis again? The hypothesis. What were you expecting from the experiment? I think it's in one of the first slides. The first few, like the one or no, I think it's second slide maybe. This one. So what were you trying to use the bacteria cellulose for again?
In other research, they always use to such as medicine and the other, like absorbing oil on the sea and trade of certain bonds and treatment of burns. Yes, and making composite materials to trap heavy metals in the water.
Okay. Okay. So you're trying to find a cheaper way to produce bacteria cellulose, right?
Yes.
Okay. And is the structure from? I know the pineapple and the banana makes different density of the bacteria cellulose, right? What about? Have you compared it with the one from the tissue culture, from the one from the tissue culture technique? If the structure is different, so banana pineapple versus the tissue culture.
It has some difference, and when we make the bacteria cellulose in tissue culture, it always use the bacteria food. Yes, I don't know what's name about that. Yes, don't try to use about the banana or the pineapple. Yes.
I'm just trying to see if the property of the bacteria cellulose will be the same if you make it with the agricultural leftovers, because those things are normally used. Those things are being implemented because they're made by the tissue culture, right?
Yes, yes.
So if you change the recipe, use the agricultural leftover instead, I don't know if the properties will be the same. Do you know?
It's same. Yes. It have same properties.
So you try to use the SCOBY, or you try to make the SCOBY?
We try to make
So
SCOBY.
you try to make the SCOBY. So SCOBY is the bacterial cellulose. Is that correct?
Yes.
Okay, so you use different carbon sources and try to make the SCOBY or the bacterial cellulose, and then you characterize the properties of SCOBY. Is that correct? Okay. So what are you going to do with the SCOBY next after you get a very good one? So if you characterize, you look at the water retention, you look at the dry weight, you look at the structure. What will you do next with this SCOBY? Yeah.
We would like to go to use to absorbing oil.
Absorbing oils?
Yes.
Oh, okay.
It has some properties to yes, can make the ค่ะเอาไว้มีเขาจะมีแบบเอาไปดูดซับน้ำมันอะไรอย่างเงี้ยค่ะ
คือสโคบี้มันเป็นส่วนซึ่งเหลือจากการหมักคอมบูชาอาจารย์เข้าใจถูกไหม
ใช่ใช่
ก็คือมันจะเป็นก้อนๆจากแบคทีเรียหรือจากเศษเซลลูโลสที่เหลืออะไรเงี้ยเนาะก็กลายเป็นแผ่นขึ้นมาดังนั้นไอเดียก็คือจะเอาตัวนี้ไปแอบซอบน้ำมันใช่ไหมออยล์แอบซอบนั้น
เขามีแบบเอาไปผสมกับแม่เหล็กค่ะแล้วก็เอาไปเพื่อที่จะไปดูดซับน้ำมันใช่ค่ะมีรีเสิร์ชไปแบบ
มันมีข้อดียังไงทำไมมันสโคบี้ที่มีแบคทีเรียมีเซลลูโลสสามารถดูดซับน้ำมันได้
คือมันเป็นเซลลูโลสที่เกิดจากแบคทีเรียใช่ไหมคะแล้วมันก็จะมีขนาดรูพุนแบบขนาดนาโนอะค่ะมันจะมีขนาดรูพุนที่มากกว่าแล้วก็จะสามารถดูดซับน้ำมันได้มากกว่าค่ะแล้วก็ใช่ค่ะมีคนไปวิจัยแล้วก็ลองทำค่ะแล้วก็พบว่าเขาสามารถดูดซับน้ำมันได้มากค่ะใช่ค่ะแล้วก็อีกทั้งแบคทีเรียเซลลูโลสเป็นวัสดุที่แบบไม่มีเขาเรียกอะไรคะแบบเป็นวัสดุธรรมชาติค่ะก็คือไม่มีอันตรายต่อธรรมชาติเนี่ยค่ะสามารถนำไปดูดซับน้ำมันได้
โอเคค่ะโอเคขอบคุณมากค่ะ
Thank you very much
Thank you for the feedback, commentator. We would like to invite our commentator, Dr. Hattapak Desha Kaisaya, onto the stage to give the certificate of presentation.
The last presenters are from National Institute of Technology Kosen Tsuyama College, and their project title is Effects of External Factors on Body Color Change of Tree Frogs. Please come on to the stage.
Hello everyone, my name is Rei Nakagiri. My name is Nijinosuke Kojima. We are from National Institute of Technology Tsuyama College. We are going to talk about effects of external factors on body color change of tree frogs. This is a Japanese tree frog. It is relatively small frog, about 2 centimeters. They have 3 types of chromatophore. In the skin. The tree frog then uses the hormone to move it. It is generally believed that tree frogs change their body color to match the of their surroundings. However, they can exhibit completely different body colors in the same location. There may be more than one factor that influences the body color change of tree frogs. Objective 1 to investigate the effects of color change in the surrounding environment on body color change. To investigate whether there are differences between regions in individuals from Klasky City and Siama City, which are about 60 kilometers apart. This is Siama City, this is Klasky City. Methods. 1. Stick pieces of drawing paper on an empty breeding case, all side and floor. 2. Places petri dish and water in breeding case. Part 3. Other frog and waited 2 days. 4. After 2 days, take a picture of a frog. 5. 9 random points were selected from the photos. RGB values were measured and body color was determined. 6. Perform the same operation with other colors. This was done in the following order: blue, purple, black, orange, green, yellow, and red. Look at this table. This color line is drawing paper's color. This line and individual 1 and 2 are from Klasky City line. 7 and 8 and 9 is Siama City from Siama City line. While different regions show similar body coloration, they may also show different body coloration in the same region. Individual 1 and 7 and 9 similar colors. 1 and 2 different colors. It is highly likely that regional dispersion do not exist at dispersion as great as those between Klasky City and Siama City. So I focused on white. Um, figure 1, RGB value of individual A. Blue wine is component wine. Red wine is red component. Green wine is green component. Yellow wine is half of blue and blue, red, green totals. We get to what to drawing paper. In each component, the changes are different, but when one looks at the total, the changes are similar. Yellow wine. And drawing paper individual 8 is similar yellow wine. Yellow wine is point is higher, its color is whiter. Lower is color darker. And brightness may have an effect on body color changes in three flugs. Conclusion. Ambient brightness may be related to body color changes in three fog. Thank you for listening.
Thank you for your excellent presentation. Next, I invite our commentator to give advice or comment about this project.
Thank you. So that was an interesting presentation on the three frogs. Why did you choose to randomize the area of the color that you're going to measure? Like, why random? The 9 points. Why random? Why not the same place in every frogs? And also, how did you randomize? Why and how?
Average. Frog body color right. Little different. So we take average 9 point.
Okay. Randomly select 9 places and then just average them together. Okay. And in that cage, is it complete? How do you control the other environments? Like, you put paper there, right? You put a drawing paper, which is colored paper, right? Colored paper, and the other sides, is it dark? Is it black? Or is it transparent? Can you see through it? Can the frog see outside the cage? No. Okay, just the drawing paper. Okay. and the light from above. Okay. Mm. And do you keep the light, like the light is always on, right? Mm. What about the sound? Does this prevent the sound from going in? Sound like, mm. Can the frog hear things inside? Mm. Do you have any hypothesis? Why do you think they change color? For what purpose? Like why do they change it? Camouflage. Mm. Okay. So there's no different, right, from the result. No different between cities. No different, right? Is there any different between between the two cities? Frogs from the two cities. Is there different? Conclusion. So brightness affects, but you tested two cities, right? From two cities, is there any difference between frogs from these two places? So frog from what was go the map cities. Yeah, frog from Suyama and frog from Kurashiki. Anything different? No. Okay. How did you know it's the brightness? In your conclusion, it says brightness. How do you know the brightness affects the change if there's no different? You conclusion. In conclusion, you said brightness, right? Yeah, brightness. Yeah. Mm. It's all right. I'm sure you have an interesting reason. It's all right.
Thank you for the feedback, commentators. We would like to invite our commentators, Associate Professor Dr. Vilailak Siriphorn Dunasin, onto the stage to give the certificate of presentation. Well, ladies and gentlemen, we have come to the end of the sessions. Thank you to all presenters, honorable commentators, and participants. We hope you enjoy the reminder of Thailand-Japan Student Science Fair 2023. See you again in 2025 at Princess Juraporn Science High School, Pathum Thani. Goodbye. Everyone, please come on to the stage for take a photo group.